So, who exactly are the key players in immunity? Well, the first one is the pathogens. And what do I mean by pathogens? I mean bacteria, viruses, parasites, and even prions, which are protein infections that work kind of like a virus in a lot of ways. Who are the good guys? These are your immune cells, your white blood cells, and they come in a lot of different flavors. Some of them are involved in that innate immunity. That means that they don't change what they do based on the strain of virus or whatever. The response is kind of always the same sort of thing. They have one job and they do that, and it doesn't mean it doesn't work against the pathogen. It just isn't adapted to it. And then you have cells that adapt. This means that they mutate in ways that enable them to better respond to that specific pathogen. And we're going to talk about each type of cell on its own. You have multiple different kinds of white blood cells. And each of these has a slightly different way that it handles pathogens. So I'm going to start with the neutrophils. Neutrophils consume bacteria through a process called phagocytosis. Phage means eat, cyto refers to cells. So basically it means eat cells. And that's what they do. They kind of go up to these cells and they engulf them. And inside of the neutrophils, there are enzymes which will start to digest the pathogen. Macrophages work in a very, very similar way, where they are going to also engulf the pathogen and digest it. We'll talk in a little bit about what they do with adaptive immunity because they have a function over in that realm as well. You also have eosinophils. What eosinophils do is they go up to parasites and they basically dump the enzymes from inside of the cell on parasites. Eosinophils and basophils are cells that are often referred to as granulocytes because they've got a granular appearance. Those granules are actually vesicles of enzyme, which they kind of barf out onto, in the case of an eosinophil, a parasite. What basophils do is basophils, again, barf out their granular they barf out their vesicles, and those vesicles are going to cause inflammation. While this may sound like a bad idea, remember that inflammation increases blood flow to an area, and in the blood there are going to be more white blood cells. So basically what it's doing is it's bringing more white blood cells to the area where they're needed. In addition to those, you have natural killer cells. They do kind of what it sounds like. They kill things. And what natural killer cells do is they have molecules that are signaling molecules that they release that are called perforins. And if you think about something perforated usually is made so that you can tear it. It's got little holes in it. They secrete these perforins onto the pathogen, and the perforins create little perforations in the cell membrane. Well, if the cell can't keep its insides inside and its outsides outside, it dies. So the insides of the cell leak to the outside and the pathogen is killed. Now, all of these responses will work against a wide multitude of pathogens. Some of them are a little more effective against something than others, and it doesn't really matter exactly what the pathogen is. Eosinophils are better with parasites, but it doesn't matter if that parasite is a schistosome or if it's a tapeworm. They don't really care. They're going to combat everything in a very, very similar way. So let's take a second and look at what they're doing. So here's an example of you having some sort of really horribly infected cut. In that, the pathogens are going to come in through that cut. They could be bacteria. Those macrophages are going to travel through your bloodstream and kind of migrate out and start to eat those pathogens. While they're doing that, you're also going to have dendritic cells doing the same thing, natural killer cells dumping perforins all over the pathogens and killing them, basophils releasing inflammatory compounds like histamine to bring more blood to the area, and eosinophils will also be releasing compounds to try to get cells to migrate there and try to fight off the pathogen. So one thing people love to do is they love to always take Tylenol if they have a fever. And it turns out you actually shouldn't do that, because the fever is your body sort of getting your immune system ready and trying to kill off that pathogen. Now, does it mean you should never take Tylenol or Advil for a fever? Of course you should if it's making you miserable. But if you are dumbing down your immune response by using those drugs, then you're kind of prolonging the disease. So next, what I want to do is start to talk about the adaptive cells and how they adapt. These are your lymphocytes. So this is going to be B and T cells. Natural killer cells also fit into the lymphocyte category, but for my purposes, I kind of put them, I put them in the category with innate immunity, because that's kind of where they go. Now let's take a closer look at those B and T cells and why exactly are they called adaptive immunity. B cells secrete something called antibodies, and these antibodies actually get adapted so that they best bind to that specific pathogen. What do I mean by that? Well, when you get sick, what's going to happen is your B cells are going to go through a rapid period of hypermutation. During that rapid period of hypermutation, they're going to be tested as to how well they bind to the receptors on the outside of that pathogen. Remember, those are the receptors that show it that it is not your cell, it's something else. And whichever ones bind really well are kept and they are cloned. Whichever ones don't bind well die off. And what your body keeps doing is it keeps trying to find better and better and better B cells at binding this pathogen. Once those antibodies or those B cells create really good antibodies, those antibodies start getting released. And when those antibodies get released, they bind to the receptors on the outside of those pathogen cells. And they basically stick up like a little flag on that pathogen saying, hey, if you're another kind of immune cell, come over and kill me. Or what they'll do is they'll surround the pathogen cell so that it can't infect any other cells, because its entire membrane is just engulfed, encapsulated by these antibodies. So one thing you may wonder is what happens, like how do the B and T cells, those adaptive cells, get activated? It turns out that your macrophages will engulf the pathogen and put part of that pathogen into their cell membrane. When they do that, they go to the B and T cells and they present it. It makes them something called an antigen presenting cell. And they show that to those B and T cells. And when they show it to the B and T cells, it activates them. I like to think of this like in those movies or Game of Thrones or whatever, where they bring the head of the invading army to the king and they're like, hey, is this the bad guy you're looking for? Because that's basically what those macrophages are doing. And then those B and T cells are checking it out and they're saying, yeah, that is the guy we're looking for. And then they launch their attack. When we look at T cells are a little bit different. There are two flavors. There are helper Ts and killer Ts, or cytotoxic Ts. So these cells also, like your B cells, go through a period of rapid hypermutation where you try to make T cells that best bind the pathogen, but instead of shooting out something like an antibody, which explodes out of the B cell and goes to the pathogen and binds it, now those T cells are directly binding with the pathogen using their receptors. So it's still on the cell, it's not being shot away. The killer T cells or cytotoxic T cells, when they bind to that receptor, they shoot out something called cytokines. Cytokines, an example, would be a perforin. So something that causes that specific pathogen to self-destruct. What helper Ts do is helper Ts also bind to the pathogen, But once a helper T binds to the pathogen, it recruits B cells to start making antibodies, and it recruits T cells, killer T cells, to start coming to the area. So the helper T cells kind of turn on the Bs and the killer Ts. One of the things that happens in immunity is if you look at people that have HIV, their helper T cells are getting killed by the disease specifically. If those helper Ts are killed, now you can't activate the regular B cells, so you can't really make antibodies very well, and you're not activating the killer Ts, which are going to kill off the virus. This is why people with HIV typically die of common ailments such as the common cold. It doesn't have to do, they usually don't die of AIDS. They usually die of a virus that they can't fight off because their adaptive immunity is basically imploding. Once you have B and T cells, which are really good at fighting off your specific pathogen, some of those cells will get cloned and put into something called immunologic memory. This immunologic memory is basically like some storage of these cells so that next time you get sick with the same virus, you already have cells that will win this immune challenge, and they can recognize that pathogen right away. So you don't have to wait until a cell recognizes the pathogen. What this means to you is your body may fight off the disease before you ever notice you're sick, or it could be that you're sick for a much shorter period of time. When we get vaccines, the reason that vaccines work is that you're exposing the body to a very small, weakened, or dead virus. That small, weakened, or dead virus will trigger the immune response and trigger this period of hypermutation in those B and T cells, and it will create Bs and Ts that recognize the virus, but it does it without getting you sick. Now, the reason why sometimes you have to get boosters or sometimes you don't develop immunity is sometimes you fight off the virus before you can finish that hypermutation. So this is why we often have to get additional shots to make sure we have immunity, or we know that after a period of time some of these memory cells do die off, and so you lose your immunity and then you have to boost it up again. People love to talk about the flu shot and the effectiveness of the flu shot, and it turns out that what's going on at the CDC when they make a flu shot is they're looking at what are the strains that they think are the most likely to come out that season. Sometimes they get it wrong, and sometimes what happens, and by the way, I mean, they're doing this with a lot of data, and there are a lot of strains that are going to come out. So sometimes they do get it wrong, and sometimes what happens is you don't develop the immune response. Maybe when you go in for your flu shot, you have a fever, and if you have a fever, your immune system is already reacting, which means it kills off that virus before you get really good binding B and T cells. Sometimes they weaken it too much, and again, it dies off before you get those good B and T cells. So sometimes it just doesn't cause the immune response that they're hoping it will cause. And if it doesn't cause the immune response that they're hoping it will cause, then that means that you don't get the immunity to the flu. As a result, that means that sometimes the flu shot isn't effective against the current strain, and this does happen from time to time. Now, in our discussion of immunity, I would be totally remiss if I didn't mention herd immunity and what that means. The idea of herd immunity is that there's a certain percentage of the population that is going to get sick. So if you come in contact with someone who's sick, you can get sick. Different diseases have different transmission levels. One of the reasons we're so worried about corona is that corona has a very high transmission level. So what ends up happening is those people will get sick and they'll develop their adaptive immunity. This can also happen through vaccines. And what that creates is that creates a portion of the population that cannot get the disease. So a portion of the population that is essentially immune to getting it and cannot spread it. What we want to do is we want to create insulation around the people that do not have that immunity. And by making a certain portion of the population immune, and it would probably surprise you, but usually it's at least 85% of the population has to be immune, then we insulate the people who can get the disease. Maybe the vaccine didn't work on them for whatever reason. We insulate them from the people who are actually sick. And that's why it's called herd immunity. Vaccines aren't necessarily about you not getting sick. It's about you not transmitting that disease to others and providing that insulation to people that maybe can't get the vaccine because they have an egg allergy or they're undergoing chemotherapy or something like that, and they can't get that vaccine. You are providing them with insulation because you are putting one more person between them and the person who's sick. You may be wondering what this has to do with blood type, and I'm going to cover that in the next video.